Good morning, children. Welcome to your biology class. We were discussing about Mendel's experiments in garden pea plants. I believe you understood what is meant by monohybrid cross. So today, let's discuss about dihybrid cross. Children, you learned about monohybrid cross. In monohybrid cross, Mendel selected mono means one. So only one pair of characters he selected. But in dihybrid cross, he selected two pairs of characters. Here, he selected the shape of the seed and also color of the seed. Shape of the seed and color of the seed he selected for his dihybrid cross. So, the cross is like this. So, one parent is having round green seeds. Round, the shape of the seed is round and the color is green. And other parent, second parent is having wrinkled yellow seeds. Wrinkled is a shape and yellow is the color of the seed. Clear? So here you can see two characters, shape and color of the seeds. So that this cross is called as dihybrid cross. Among the pair of characters, one is dominant and one is recessive. Dominant, we will always represent dominant characters in capital letter. Dominant alleles in capital letter. So here, among the round and wrinkled shaped seeds, round shape is dominant over the wrinkled one. So he represented the round shape in capital R. Dominant, it is dominant. So it is capital R. Capital R, capital R. The recessive one, wrinkled, that is small r, small r. Okay. Then two colors are the green and yellow. Among the two colors, yellow color is dominant over the green color. Okay. So he represents these colors in the letter Y. So here yellow color is represented by capital Y because it is dominant color. Here green is represented by small y. So round green will be represented as capital R, small y, and wrinkled yellow by small r, capital Y. Then he crossed these parents. Okay. Then how can we cross? First is the allele formation. So here two characters are there. Round is represented by the alleles capital R and capital R. The same alleles are there. So we have to select only one from that is capital R. Okay. Here green represented by small y alleles. Same type of alleles are present. So we also select one small y from the pair. Then capital R from here small y. It forms capital R small y allele from the first parent. And here small r that represent the wrinkled character, isn't it? So here also same alleles are there. So we select one small r from the wrinkled genotype, and here capital Y. From capital Y we select one capital Y. So it forms small r capital Y allele. Okay, small r capital Y allele from the second parent. Okay, so to generate F1 generation, to produce F1 generation, we cross these alleles. So what will be the genotype of the F1? Here capital R small y and here small r capital Y. So we have to write capital R and small r together. Okay. So capital R small r, then capital Y small y. Capital letters we have to write first. Okay. So capital R small r and capital Y small y. That is the genotype of the F1 generation. So what will be the phenotype? What will be the character of the F1 generation plants? So here capital R is present. Capital R denotes the round shape. Right? It's a dominant yellow color. So all the seeds of the F1 generation were round yellow seeds. Okay, then next step is the F2 generation, production of F2 generation. How? By self-pollination. Self-pollination means F1 crossed with F1. So what is F1 here? Capital R, small r, capital Y, small y. So capital R, small r, capital Y, small y cross with the same genotype, that is capital R, small r, capital Y, small y. So how can we make alleles? Here some complication is there because two types of characters, two types of alleles are there, right? So the first genotype is capital R, small r, capital Y, small y, right? So here capital R, small r, two types of alleles are there, and here also capital Y, small y, two types of alleles are there also. So we have to form the alleles like this. From the first pair that denotes the shape of the seed. Okay. So from that we have to select the first one, that is capital R. Capital R cross with capital Y. That forms capital R, capital Y allele. Likewise, capital R with small y, that forms capital R, small y. Then capital R is over now. Then next one is small r. So small r with capital Y. So small r, capital Y. Then small r, small y. These are the four alleles produced from the F1 generation. So the same type alleles will be produced from the second parent also, because we done self-pollination, F1 cross with F1. So here also the four alleles, and here also the same four alleles will be there. Okay, then we have to cross these four alleles together. How can we cross? So we can use Punnett square for this purpose, because the number of alleles are more when we compare to the monohybrid cross. So this is the Punnett square. You have to draw a table like this to do the dihybrid cross. To produce the F2 generation of the dihybrid cross, because the number of alleles from the first and second parents are more when we compare to the monohybrid cross. Here 4 is there, here also 4 is there. So total 8 alleles are there. Okay. So the first one, it represents male. This one is the male, symbol of male, and here it is the female. That is two types of parents. Okay. So the alleles of the first parent you have to write here. Capital R, capital Y, you can see. Okay, capital R, small y, small r, capital Y, small r, small y. And here the alleles of the second parent you have to write. Okay, capital R, capital Y, capital R, small y, then small r, capital Y, then small r, small y. Then how can we cross? So the first alleles of So finally you will get this result. So these are the genotypes and phenotypes produced in dihybrid cross in F2 generation. Next you have to find out the phenotypic ratio for dihybrid cross. How many round yellow plants are produced? 9. Then round green? 3. Wrinkled yellow? 3. Wrinkled green? 1. So the phenotypic ratio will be 9:3:3:1. In dihybrid cross, you don't need to find out the genotypic ratio. So this is an important question. Practice well. Okay. Our last topic is sex determination. How is the sex of the new individual determined? Different species use very different strategies for this. So let's discuss the strategies of different organisms for sex determination. In some animals, like a few reptiles, the temperature at which fertilized eggs are kept determines whether the animals developing in the eggs will be male or female. In some animals, such as snails, individuals can change sex, indicating that sex is not genetically determined. In human beings, the sex of the individual is largely genetically determined. In other words, the genes inherited from our parents decide whether we will be boys or girls. In the case of human beings, XY is the male chromosome and XX is the female chromosome. Male can produce sperms as their gametes. A male can produce millions of sperms at a time. So 50% of the sperms containing X chromosome and the remaining 50% containing Y chromosome. Female can produce ovum as their gamete, but female only produce one ovum in month. That ovum containing only one type of chromosome, that is X chromosome, because the female is having XX chromosome. So in ovum, only one type of chromosome is present, that is X chromosome. So at the time of fertilization, if the X containing ovum fertilized by the X containing sperm, the result will be the zygote which is having XX chromosome. Isn't it? Then what will be the sex of the child? It's a female baby. Then if the X containing ovum is fertilized by the Y containing sperm, then the zygote is having XY chromosome. Then what will be the sex of the baby? XY is male. So the child will be a male. Children, you have to do this dihybrid cross in your notebook. I also attach the YouTube links of our second practical, binary fission and amoeba, budding in yeast and hydra. Watch the videos and learn the theory, diagrams, observations and prequation of the practical. We can discuss more questions in our revision session. Thank you for listening me. Thank you.